Fibroblast growth factors, or FGFs, are a family of cell-signaling proteins discovered fairly recently in 1973. Originally, they were identified as mitogenic growth factor in pituitary extracts by Armelin. However, Gospodarovich, after isolating the protein more precisely using cow brain extract, testing in bioassay, identified that the protein caused fibroblasts to proliferate. Currently, there are 22 known FGFs in mammals across 7 families, consisting of 18 proteins and 4 receptors. FGFs have several functions such as cell proliferation, migration, and differentiation. In adults, they respond to injury and tissue repair. The different functions are dependent not only on the specific FGF ligand and receptor involved, but also the activation of various signal transduction pathways. This video will focus on FGF's proliferative functions in mitogen and discuss whether these can be attributed to the activation of a specific signaling pathway. The proliferative role of different FGF proteins is seen in limb development in the formation of the apical ectodermal ridge, or the AER, a distal structure of each limb bud formed from ectodermal cells that acts as a signaling center, key in limb development. FGF10 is expressed in limb mesenchyme, but only signals the overlying ectoderm where it activates FGFR2b to form the AER. From here, FGFR2b activates the expression of FGF8 in the AER. FGF8 then signals back to the mesenchyme and activates FGFR1c, creating a positive feedback loop. Reciprocal signaling between FGF10 and FGF8 leads to outgrowth and proximal distal patterning of the limb bud. In adults, such development of the signaling pathways are reactivated in response to injury to repair tissues. The way that FGFs induce biological responses like this is through signaling via the aforementioned transmembrane receptors called FGFRs. Their basic structure is comprised of 3 extracellular immunoglobulin-like domains, a transmembrane domain, and 2 intracellular tyrosine kinase domains. Examples of the different FGF signaling pathways are the P13 kinase AKT pathway and the PLC gamma pathway, but the RAS-MAPK pathway is considered to be the main one. This is the pathway activated in the mesenchymal cells immediately adjacent to the AER during limb development. In basic terms, the RAS-MAPK signaling pathway works as follows. Extracellular FGF binds to its receptor, resulting in dimerization or the stabilization of dimers of 2 ligand-bound receptor molecules. This then activates the intracellular tyrosine kinase domains, which subsequently phosphorylate each other. The phosphorylated receptor cells then recruit 2 adapter proteins, growth factor receptor bound protein, GRB, and SONOS. These in turn recruit and activate RAS at the plasma membrane. This results in the activation of the first serine or threonine kinase in the cascade, RAF in mammals. RAF then phosphorylates and then activates the next kinase in the cascade, MAPK kinase or MAPKK. Following this, MAPKK is phosphorylated and activates MAPK, mitogen-activated protein kinase. This may then phosphorylate other kinases, such as P38, JNKs, or ERKs, or enter the cell and phosphorylate transcription factors. Thus, gene expression is activated. Several studies claim this pathway is implicated during cellular proliferation. However, pinpointing the specific role of FGF in this pathway and directly link it to cell proliferation is problematic. Attempts to determine the role of the RAS-MAPK pathway in proliferation in activating key components in the pathway through using dominant negative forms or chemical inhibitors, assessing how this alters the outcome. Several studies using this method demonstrated the pathway does indeed have proliferative effects. However, the RAS-MAPK pathway, unlike the other pathways, also mediates FGF-induced cell cycle arrest of certain chondrocytes and induces differentiation of cells called PC12. Furthermore, to relate back to the limb development example, the FGF signaling, where the RAS-MAPK is also involved in differentiation of the mesenchyme. Therefore, no simple correlation can be made between the activation of this pathway and the biological response to FGF. Multiple factors may cause this. To name a few, research suggests the strength and duration of activation, or even the quantity of certain kinases, can influence the cell response. For example, studies in Drosophila showed low levels of RAS1 protein activate expression of a gene called tailless, whereas high levels lead to the activation of both tailless and another gene called huckbine, which had implications on the formation of terminal structures of the embryo. Ultimately, whilst the activation of specific pathways may influence how cells respond to FGFs, they don't entirely explain the specific role of FGFs. It is important to remember that, rather than being linear, the RAS-MAPK pathway is part of a complex network of intracellular communication, affected by many factors. Limb development is a good example of this, since the FGF signaling is involved in both proliferation and differentiation of the mesenchyme. For this reason, to find out what will elicit a proliferative response in a cell from FGF, a holistic view must be taken that is likely to be equally intricate to the complex nature of FGF.